So, hello guys, welcome back to our channel Biolab India, and this is Vamsi. Yes, wait a minute guys, let me just connect my tab to the screen. Then we'll be starting with biotechnology principles and processes, the questions, and then we'll be moving on to the applications part. In this biotechnology principles and process, you have some 50 questions that I have gathered for you from my reference book, that is the PYQ book. If you want more after doing this 50 questions, I'll also provide you some other. I'll be now only taking some of other questions from the other books also, and then we'll be doing lots of questions today. Today is a question day. Please put your notes beside you so that you can write the answers. You can write your answers in that book. Yes. Once, let me check whether I am live or not. Please share this, guys. Please share to your friends so that they would come. I'm perfectly live and my voice is also audible. It is fine. Okay. We'll be moving on now. Come on. Let us move on. Let us move on to this practice session on biotechnology principles and processes. Yes, who are joining, they can join in the middle, no problem. Let us start with the first question right away. Yes, I hope you have revised this chapter since I have already completed the one shot on this. Starting, the first question is this question is basically from NEET 2017 PYQ. This is an PYQ, yes. Bennett. Okay, not able to write on this. Yes, give me a minute, guys. I'll just change this. I'll reopen this. Wait a minute. Yes, I have now. I hope I am able to write it. Yes, now it's fine. No problems. Okay. Let us move on. With the first question. This has appeared in 2017, and this question says the process of preparation and purification of expressed protein before marketing it is called as. So it is asking just you the term which is used for purification of a gene or a protein which has to be gone into the market. Yes, we have produced the gene and everything is completed. We have to launch it into the market, so that we have to purify the gene. We have to remove its side effects and impurities. So now what's that process called as? Yes, quick. Put your answers in the chat. Put your answers in the live chat. Yes? Okay. That process is called as downstream processing. I hope you have done it right. Downstream processing is the right answer. Okay. Why it is called as downstream processing? Open your NCERT first page only. It has been written that biotechnology provides a best catalyst. I have already told you it provides the best catalyst. It only provides the optimum conditions It only helps in downstream processing, and that downstream processing is called as your purification. Okay, that has appeared in your NEET 2017. Next one, with other, see, with other options we no need to go. We have to check the other options when we don't know the correct answer. See, there is a small trick here in order to, if you have some confusion in the questions, you do one thing. You just follow the elimination method without any looking forward for the answer. Just follow elimination method. See, if you have for suppose upstream processing, you have never heard about upstream processing in biotechnology part, so no need to consider that. Okay, bioprocessing, that also we haven't. So post production processing, this one you might get doubt between downstream processing and post production processing. Just think over it. When you are reading your NCERT, you will be reading the words, right? So then you will be your mind calculates the thing, and then it says downstream processing is the correct answer. Follow elimination method if you are not able to locate the correct answer. So now the second question, this has also appeared in NEET 2017. This is your second question from NEET 2017. So, what is the criterion for DNA fragments movement on the agarose gel during gel electrophoresis? We have seen the gel electrophoresis experiment, and on that, what is the criterion? See, the first option, larger the fragment size, the further it moves. Yes, I have already told you an example about a fat runner and a lean runner. Yes, I have already given you that example, right? Now, based on that example, please try to do this question. First, the smaller is the fragment size, the further it moves. Yes, the positively charged fragments move further. Are there positively charged fragments and negatively charged fragments in DNA? No, whole DNA is negatively charged fragments, so let's eliminate this. We have three options to choose. But they have given you that negatively charged fragments do not move, so we can eliminate this. DNA whole itself is negatively charged, and how can those fragments move? So eliminate D, eliminate C. We have two options to choose. Now get the example that I have given you between fat runner and a lean runner. Now your second option becomes your correct answer, option B. Yeah, option B. Very good going. I hope yes, no one is putting in the charts. Let me just join in the stream so that I can see those charts. A minute, guys, just give me a minute. Now I am here. One more check. Now, okay, everything is fine, nothing is happening, right? Now see, let us move on to the third question. This has also appeared in NEET 2017. This is your third question that has appeared in NEET 2017. Okay, third question. Now, let us see. A gene whose expression helps to identify transferred cells is known as. Come on, guys, I have told you this. You can select the recombinants. And kill them, and you can separate the non-recombinants. Yes, come on. Vector, plasmid, structural gene, selectable marker. It is helping you to select, so it is selectable marker, of course. You have got, if you have done these three questions correct, you have got 12 marks in your bag. Just keep moving ahead. Now this is the fourth question that has appeared in NEET 2017, and this is the fourth question for the day. Let us move on. The DNA fragments separated on agarose gel can be visualized after staining with. I have already told you that the DNA fragments after separation, they cannot be seen. In order to be in order to see them, you have to put something so that it gives some color. Then you are able to view it. So that something is very important, and that something is your question. Now see, you can apply very clearly the elimination method. See, bromophenol blue. No, we haven't ever heard about that name in the biology. Next one, acetocarmine, no. Acetocarmine and aniline blue. These three we haven't ever heard about them. So ethidium bromide becomes your answer, and that gives a bright orange color to the DNA strands so that they are visible. Clear? Yes. Very good. Now. Very good. That's it. Okay. Now let us move on to the fifth question. This is your NEET 2016 question, phase 1. Okay. The Taq polymerase enzyme is obtained from Taq polymerase. I hope I haven't told you this in my live in the session, and I have asked you to please do these parts which I have left. If you have done those parts after my lecture, you can simply and very easily answer this question. Taq polymerase. I'll give you a small hint also. Taq, Ta and Q. Those two are present in the answer also. See, Thiobacillus ferrooxidans, no. Bacillus subtilis, no. Sorry for pronouncing these wrong. I am not a good pronouncer about these spellings over. Okay, Pseudomonas subtilis, no. Next one, Thermus aquaticus. That's your answer, Thermus aquaticus. Let me give you a small hint how to remember this Thermus aquaticus now. See, Taq, T means Thermus, AQ means aquaticus. Done? Completed? Remember the polymerase. Okay, Thermus aquaticus. Yes, now it is fine. Yes, there was some net issue, so that is what. Now let us move on to the sixth question. This has appeared in your NEET 2016 phase 1 again. Now, which of the following feature is not a feature of plasmid? Plasmid features. Come on, plasmid is a circular extracellular DNA, not extracellular, like extranuclear DNA, extra DNA. It is not present in the nucleus, it is present in the cytoplasm, and it is a circular in shape, it is double stranded. Come on. Yes, it is transformable, circular shape, transformable, independent of replication. It is having its cytophory, so it is independent of replication. Then hence C will be your answer. It is not single stranded, but it is double stranded rather. Okay, this is your sixth question of the day, and it has appeared in NEET 2016 phase 1. Now let us move on to the seventh question of the day. That has also appeared in NEET 2016 phase 1. And which of the following is a restriction endonuclease? Restriction endonuclease. So easy question, so easy. Until now, if you haven't the tough questions until now are, if you haven't read the NCERT, this Thermus aquaticus question would be tough for you. Next one is like no tough questions are now. Until now there are no tough questions. Every question is easy until now. This question is very easy. No need explanation, guys. Come on. Protease. Okay, I am giving you protease. It is help to digest proteins. DNA is one. It is help to digest DNA. RNase. It is help to digest RNA. Okay. Now, which of the following restriction enzymes produces blunt ends? This is somewhat tricky. Yes, it is somewhat tricky. What are blunt ends? The ends which are not sticky. It is tricky, and the ends which are not sticky are called as blunt ends. See, blunt ends means there is no complementary between these two cut strands. Okay, there is no complementary between these two cut strands. That is the thing. I hope this is not given, not mentioned in the NCERT about this. So I don't know why they have asked this in the phase 2 question. So it is Eco R5. Okay, Eco R5. Hindri see, here also you can use the same concept of eliminating questions. See, Hind 3, no, we know about Hind 3. This one we haven't ever heard. you can get a confusion between SAL1 and EcoR5. SAL1 and EcoR5. So SAL1, okay, SAL1 and EcoR5. So just think over it. Just think over it. Where does SAL1 has its restriction endonuclease site? Okay, think over it and then put the answer EcoR5. It is not present in NCERT. No need. Just forget about this if you get wrong. Also, this is a tough question. See, questions. My our sir always tells us that questions can be tough. One reason is they are not present in your textbook. Second reason is you haven't studied at all that concept. The third reason is you are not getting that question during the examination time. So these are the three reasons for a question to be tough. Okay. A little adjustment of the camera. Now. Eighth one completed. This is a little tough question. Please leave it. This is not present in NCERT. Okay. Let us put this tough question with this. Until now, okay, after 10 questions, let us check. Now, ninth question. Which of the following is not a component of downstream processing? Not a component of downstream processing. Come on, tell me. It's very easy. Expression. It is not a component of downstream processing. Rather, it is a component of genetic modification. Yes, you put the gene there and it is getting expressed. So it is not a component of downstream processing. Downstream processing exclusively, it has the contents of purification only. It is having the contents of purification only with downstream processing. Okay, clear? Now let us move on to the 10th question. Yes, is this? Oh, sorry guys, sorry. Yes, I haven't seen that the screen is not working, or like the sharing has been stopped here. I haven't seen that. Sorry for that. I'm just connecting it again. So yes, there was a net problem, right? So because of that, the screen might have not been visible. I hope the screen is now visible to you very clearly. I hope now the screen is visible to you very clearly. Wait a minute, not now. No, it is clear. Yes. Now done. Now let us move on. Let us move on to the 10th question. 10th question. A foreign DNA and a plasmid cut by the same restriction endonuclease can be joined together to form a recombinant plasmid using. Now think about molecular glue, not molecular scissors, my dear brothers. Molecular scissors is your restriction endonucleases, my dear friends. Now think of the molecular glue, that is your ligase. Ligase is your molecular glue. I have already told you in my session. Yes, we have completed 10 questions. Out of these 10 questions, how many of you have got 9 out of 10? Okay, I'm eliminating that thing, the 8th question. So I'm putting 9 marks. Out of 9 marks on this page, how many of you got 9 by 9? Put in the chat section. 9 by 9, how many of you have got? 9 by 9, come on. I'm just spinning it. The first set of questions. I have pinned the comment. Yes, I have pinned the comment. The first set of questions is completed. 10 questions we have done. We are done with 10 questions. Okay? Now let us move on to the next page without any further ado. Come on. This one again might be a little tough question if you haven't studied your NCERT. This might be a tough question if you haven't studied your NCERT. I'll read the question for you since it is a little blur. Stir tank bioreactors have been designed for. Stir tank bioreactors have been designed for. If you haven't read NCERT, this would be a tough question again. This would be a tough call again. I have already told you I have left some concepts from your NCERT. Please read them. So if you have read, you would opt your C option as your right answer. Okay? Read, go after this session. Please write the things where you are lacking. And then after that, go back and give it a reading. Give it a reading. That's it. Enough. These are very easy questions. Okay? That has appeared in NEET 2016 phase 2. Now we are moving on to APMT 2015 questions. These would be a tricky little. Please just concentrate and answer the questions. They will be so easy. This would be tricky again, but please with concentration answer the questions. They would be very easy for you. Now see. Cutting DNA at specific locations became possible with the discovery of. This is the most repeated question everywhere. If you see the PYQs of this biotechnology principles and processes, you can see every PYQ book which you take, every reference book which you take, you will be having this question per compulsory. Okay, now the cutting of DNA has that specific locations has been possible by restriction enzymes. What would be the other answer for this, guys? What would be the other answer for this? Now, DNA molecule to which the gene of interest is integrated for cloning is called as. Yes, this is also very easy. This is also very easy. I have given a build-up of AIPMT. Nothing is there. Vector is the answer. Yes, I know that my students and my friends will not go never wrong about these questions. Now, an analysis of chromosomal DNA using southern hybridization technique does not use. This again will be a tough call for you if you haven't studied the NCERT regarding the southern hybridization. Come on, this has appeared in CBSE APMT 2014. Answer this question. First, yes, the answer is PCR. Actually, southern hybridization involves southern blotting, western blotting. Electrophoresis is a compulsory between in any process in order to separate the DNA. Next one, autoradiography means it is a visualization of DNA. It is a visualization of DNA. You stayed with the iridium bromide, right? When you do autoradiography, you will be able to know which fragment is what. Okay, that is your autoradiography. Let us move on to the 15th question. In vitro clonal propagation in plants is characterized by. It is characterized by. Please do not look at these. Please do not look at this. Not in NCERT. Just answer with the first words. The first words are never repeated, so no need, no problem. You can answer with the first words if you know the answer. Yes, PCR is the correct answer for that. That is very easy question. I hope you have been confused this RAPD and HPLC. Yes, please just remove them. Not an NCERT, just remove them. Which vector can clone a small fragment of DNA? A small fragment of DNA can be cloned by. This is in connection with rest chapters also. Okay, which can clone a small. Yes, that is your plasmid. Plasmid is the smallest thing and it can only code for a small thing. Okay. Next one, DNA fragments generated by restriction endonucleases in chemical reactions can be separated by. Very easy question. Electrophoresis. Yes, very easy question. Gel electrophoresis from past. In the past page also we have some 3 questions on this, and in this page also we have 2 questions on this, and then this is your 3rd question in this page about gel electrophoresis. Next one, The colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of. This is also a tough call for you if you haven't read the NCERT. Okay, if you haven't read the NCERT, this would be also a tough call. Okay. Now, non-recombinant bacteria containing beta-galactosidase. Okay, that first option is. Next one. Insertional inactivation of alpha-galactosidase and non-recombinant in non-recombinant bacteria. Insertional inactivation of alpha-galactosidase in recombinant bacteria. Inactivation of glycosidase enzyme in recombinant bacteria. Come on guys, tell me, this is very easy. This is very easy. Actually, what is this? Alpha galactosidase is a gene which gives color, guys. Which gives color. If you insert any gene, if you do insertion and activation of this alpha galactosidase, there is no color. So no color means it automatically represent white. So C would be your answer. C would be your answer. Recombinant DNA will impart white color only in the dark blue background. Now, the given figure is a diagrammatic representation of E. coli vector PBR322. Which one of the following given options correctly identifies certain components? Very easy. Very easy. This is the NCERT diagram. We have discussed much on this. Come on. Original restriction enzyme. Wrong. He is asking correct, right? Yes. Correctly. No. Reduced osmotic pressure. Really? Rock means that? No. HIND3, EcoR1 are selectable markers? No. Now, HIND3 and EcoR1 are not selectable markers. They are the restriction enzyme sites. So, AMPAR and TETAR are the antibiotic resistant genes. Yes, AMPAR is ampicillin resistant gene and TETAR is a tetracycline resistant gene. Now, next one. PCR and restriction fragment length polymerization are the methods for. Come on guys, in which PCR is used? Just think for that. In which PCR is used? Gene, it is in the study of enzymes. No, to study the enzymes, there is no need of amplifying the genes, right? There is no need of amplifying the genes. Next one, genetic transformation. No need about PCR. PCR is only related to amplification. DNA sequencing, not at all. Genetic fingerprinting. Yes, DNA fingerprinting or genetic fingerprinting are one and the same. A single stranded nucleic acid tagged with radioactive molecule is called as probe. Okay, it is called as probe. An example for probe is VNTR. Okay, variable number of tandem repeats. That is an example of probe. Next one. Which one is a true statement regarding DNA polymerase in PCR? Which of the following is true for DNA polymerase in PCR? This would be again a tough call if you haven't read your PCR. Okay? Yes. Come on. Yeah, DNA polymerase, here it is in the sense Taq polymerase only. Taq, the Thermus aquaticus bacterium, which is obtained, that Taq only is this. Yes, it is highly active at high temperatures. That is your answer. Taq polymerase is active at high temperatures. How to again remember this? Very simple. Taq T temperature. Temperature is high, active. T means temperature, A means active. Which temperature? High temperature. Okay. Low temperature, we don't have concern about anything. If it is in low temperature, it is not concerned. So Thermus aquaticus, same bacteria name is saying that I am stable at high temperatures. I am active at high temperatures. Okay. The transformation For transformation, microparticles coated with the DNA are bombarded with a gene gun are made up of. I have already told you this. I have already told you this. They are made up of. Come on, come on, come on, come on, come on, come on. Gold and tungsten. I have already told you this. They are made up of tungsten. Okay. Now we have completed the second page. We have completed 23 questions. In the last page 10 questions and in this page 13 questions. Now tell me, out of these 23 questions, how many of you got all 23 correct? Okay, all 23 correct. How many of you have got? Okay, 23 questions completed. Now let us move on. Let us move on. Minute, yes. Yes. 24th question. Given below is a sample portion of DNA strand giving the base sequence on the opposite strands. What is so special shown in it? What is this? What is this? What is this? I'll not shout again. Come on, answer this. Replication completed. Deletion mutation. Start codon at 5' and palindromic sequence of base pairs. It is a palindromic sequence of base pairs. Check G, A, T, T, C, and this is the restriction enzyme nucleus. It is a site for cutting of restriction endonuclease EcoR1. Yes, it is cutting between GA. It forms two sticky ends, so not blunt ends. Clear cut, done and dusted. Okay, very easy that question was. Now, I don't know why these people ask this much lengthy question and the answer is only one line, and if you read that one line of the question, which of the following, you'll get the answer. I don't know why they write this much of story for that. Let us change, guys. Let us change this. What is this? Okay. There is a restriction endonuclease called EcoR1. What does co-part represent? I have told you in the nomenclature part, co-part represents coli. It is your species. It is your species. Now let me ask you a question modifying this. There is an endonuclease, restriction endonuclease called as HIND2. HIND2. What does D represent? Yes, D represents its strain. Okay, D represents its strain. What does IN represent? Hemophilia, influenza, guys. Influenza species. So influenza is your IN. Okay, clear, correct? Come on, let us move on. Coli is the answer. We have to complete these 50 questions and we have to move on to the 24 questions of your applications part. No problem. Agarose extracted from the seaweeds is used in. Very easy question. We are in the year of 2010, guys. Which one of the following is used as a vector for cloning genes into higher organisms? This is a question that you have to think, but no need. Higher organisms are obviously animals. In order to code for animals, we use retroviruses. Okay, clear? No. Restriction endonucleases are the enzymes which see. This question has repeated how many times? It is showing variable number of tandem repeats here. See, 2010, 2006, 2002, 2001, 1998, 1995. These many times this question has repeated, and if you answer this question wrong, it is so bad. So let us answer it correct very clearly. It is asking the definition of restriction endonucleases by giving you 4 statements. Make cut at specific positions within the DNA molecule. A minute, guys. Give me a minute. Just a minute. Okay, guys. My mother was talking with me, so I have. Okay, guys. Okay, still there is one only watching. What is this, guys? You are not sharing the session at all with anyone. Very sad. You are not sharing the session with anyone. That one is me only. You are not sharing it. Please share. Okay. Makes cuts at specific positions within DNA molecule. Recognizes specific nucleotide sequence binding of DNA ligase. Restrict the action of DNA polymerase. Remove nucleotides from the ends of the DNA molecule. No. Please answer this question correctly. This has appeared many a times. Yes, here is your answer. Here is your correct. Next one. Stirred-tech bioreactors have been designed. For this one, we have already done. We have already done this one. Yes. What is the answer? Tell me. We have already done this one. This question has been repeated twice. Once in AIPMT 2010 and I hope in NEET 2016. 2 times repeated. Watch. Next. Which one of the following is commonly used for the transfer of foreign DNA into crop plants? Very easy. Agrobacterium. Tumefaciens. Tumefaciens, no need to worry. Agrobacterium is the answer. Completed. Now, polyethylene glycol method is used for polyethylene glycol method is used for. Come on, answer the question, guys. Answer it correctly. Bang. Okay, that is gene transfer without a vector. Okay, it does gene transfer without a vector. Next one. The genetic deficiency defect the genetic defect ADA deficiency may be cured permanently by. This is a question of your biotechnology applications of biotechnology in medicine. Come on, let us solve this question. Come on. I don't know why they asked this question in biotechnology principles and process part. Come on. Until you answer, I will be doing like this only. Okay, no problem. No one is there to answer, so I am answering this question. Periodic infusions, now administering, now introducing, now enzyme replacement, now. No answer is present, guys. No answer is present. Why no answer will be present? Periodic infusions of genetic material, actually, it is not permanently curable when it is done in adult stages. It requires periodic infusion. They have itself, they told periodic infusions. So that is why this is your correct answer. You can go with it. If at all they haven't mentioned this periodic infusions word, and this would not be a correct answer, because I have already explained in the session. Go watch that session. I will not tell you now. My throat is drying. I am having cold. Okay. Gel electrophoresis is used for separation of DNA fragments based on. How many times will this gel electrophoresis come, guys? How many times? This is your, if I count, this is your some 10th question. 10th question itself on gel electrophoresis, not old. The linking of antibiotic resistance gene with plasmid vector became possible with. Again, this question is also a little tricky in the options. Okay, that is why I am doing like this. This question is a little tricky with the options. Okay, if you are my student, you will not do that. You will not go wrong with this question. The English that has been used in the question is a little tricky. English that they have used in the question is a little tricky, not the question, guys. They are asking linking of antibiotic resistant gene with the plasmid vector. So your answer goes like this. How many of you have put this endonuclease as answer? How many of you have put this endonuclease as answer? How many of you guys? There is no one to tell. Okay, put in the comment section below. That also no one will say. Okay, I know that also no one will say that. Okay, no problem. Let us move on to the 35th question. Restriction endonucleases are present in the mammalian cells for degradation. Okay, they are used in genetic engineering for ligating. What? Okay, what is this? Ligating? No, they are not molecular glue. They are molecular scissors. So let us remove that. Next one. Are used in vitro DNA synthesis? Not at all. I hope they are synthesized by bacteria and a part of self-defense. I have already told you that when a virus particle injects its DNA or genes into the bacteria, there are two enzymes released by that bacteria which are called as E1 and E2 for suppose, and this E1 cuts the DNA of virus. But why the bacterial DNA is not cut? Because the E2 enzyme adds methyl groups to the bacterial DNA in order to stop it from cutting. Okay, next one. In transgenics, expression of transgene in the tissue is determined by. Now tell me this question, yeah. Now tell me this question. This question not in NCERT, in other chapter release. Okay. No problem. It is in the molecular basis of inheritance chapter. No problem. Then also you can answer it since you have completed whole of your syllabus, right? You can answer that question. Now, the TI plasmid is often used for making transgenic plants. This plasmid is found in. Where is TI plasmid is found? Come on, tell me. Where is TI plasmid found? In Agrobacterium. This TI plasmid itself induces diseases into the plants. Okay. ELISA is used to detect viruses where the key reagent is. Where the key reagent in ELISA is. This word is not in NCERT but is in application of a line which is present in NCERT. That ELISA works on the principle of. Yes, it works on the principles of interaction between antigen and antibody and antigen interaction. So therefore alkaline phosphatase becomes your answer. That is your 38th question. Let us move on to 39th question, and we are at the end. And now we have completed 38 questions, right? Now tell me how many of you have got all 38 correct in the comment section below. I know there is no one, so that is why I am asking you to put in the comment section only. Yes, we have completed 38 questions, right? How many of you have got all 38 correct? Put in the comment section, okay? Not in the chat. There is no one. No. Let us complete this chapter. This next chapter I will be doing a part 2 video, guys. Since it is much time is already. I hope it is 1 hour. I have started the live. Since I have started the live. Yes. You have to eat. I have to eat now. So that is why. Let us do that one in the second part. That just only takes 15 minutes of our time. That is very easy chapter, right? Okay. Now let us come back to the biotechnology. Sorry guys, sorry. Yeah, manipulation of DNA in genetic engineering because became possible due to the discovery of. How many times would this fellow ask these questions, guys? I don't know. I'm not sure, pretty sure how many times this question is being asked. This is your fifth question, guys. Fifth question on same thing. So restriction endonuclease. Without restriction endonuclease becoming discovered, you cannot cut the DNA, you cannot paste the DNA using DNA ligase. So do not opt a DNA ligase, but opt restriction endonuclease. If restriction endonuclease is not present, you can go for DNA ligase. Okay. Actually, wait. A mutant strain of T4 bacteriophage R2 fails to lyse the E. coli, but when two strains R2X and R2Y are mixed, then they are lyse the E. coli. What may be the possible reason? What may be the possible reason? This question is linked with the molecular basis part, and if you haven't completed that, you could not answer this question, since about cistrons we will be discussing in that molecular basis chapter. When it comes, I'll be explaining you this question definitely. I'm just taking it a screenshot. Wait, I'm taking this question a screenshot so that I can tell you when that molecular basis comes. A small screenshot is completed. Let us move on. In bacteria, plasmid is very easy question. See my face expressions. It is very easy. My face expressions are changing whenever I say these questions. Plasmid is again the same question. What is a plasmid? Tell this fellow. This fellow is not knowing. It is a fragment of DNA which acts as a vector. Yes. Plasmid are suitable vectors for the gene cloning, because why are we choosing plasmid as cloning vectors? They are having features of cloning vectors. Here in these four options, what are the feature? Which is the feature of cloning vector? Award. Tell me. Yes. B is the feature of cloning vector, since it is having its own site of replication called as ori. Yes. Origin of replication. I don't know why my video has stopped for a moment. Okay. No problem. Let us move on to the 44th question. The process of replication of plasmid DNA other than initiation is controlled by. Other than initiation, the process of replication of plasmid DNA other than initiation is controlled by which gene? Initiation to mitochondrial. We haven't discussed about mitochondrial gene. We have only plasmid gene. Initiation to plasmid gene will do, but they have asked other than initiation, right? So you have to opt bacterial gene as your correct answer. Bacterial gene proceeds this plasmid further after the initiation. Initiation could be done by the ori in the plasmid. Once ori has completed its initiation, the replication starts, and the bacterial gene itself replicates this plasmid all over itself, and it produces large number of genes. Come on, last four questions. Let us complete within 4 minutes. Come on. Which of the following is related to genetic engineering? Which of the following below is related to genetic engineering? Mutation, so it is a natural process. Plastid, what is this plastid? It is present in plant cells, not at all required. Heterosis, we haven't never heard the word. Plasmid is the word which we have heard in biotechnology, so we put it. Now, recombinant DNA is obtained by cleaving the pro-DNA by. Actually, pro-DNA is nothing but it is non-recombinant DNA. Put pro-DNA aside and write a non-recombinant DNA. The first DNA if this is the first DNA, we do something, we do add something and then remove this and then we put another gene and we make it as an rDNA called as recombinant DNA. Okay, and what is that we are doing here? What are the things which we are using in order to cut the DNA? Yes, restriction endonucleases. This is your 6th question on restriction endonucleases. 6th question, guys. Last 2 questions, 2 minutes. Genetic engineering is possible because how many times would you ask this question? This is your 11th question on the same note. No. The phenomenon of transduction of bacteria is well understood. What is transduction? What is transduction? Transduction is a process in which genetic information of bacteria is transferred. Okay. Now, we can see the DNA by electron microscope. No, we can cut the DNA at specific sites by endonucleases like DNase 1. DNase 1 digests the DNA itself. Then how can you cut it? Restriction endonuclease purified from bacteria can be used in vitro. This is your correct answer. Now let us move on to the last question of the day, last question of the session, last question of biotechnology and processes. That is your 48th question. See, 2 bacteria are found to be very useful in genetic engineering experiments are Escherichia and Agrobacterium. Sorry. Escherichia means your Escherichia coli, plasmids. Yes, and Agrobacterium for plants, infecting plants because it is having Ti plasmid in it. Yes, it is having Ti plasmid in it. Now, we have completed, guys, this session. We are done with the session. We will be doing this biotechnology applications in the next session. Okay, next session. So we will be doing this applications not in the next session, most probably after the next session, because the next session would be on your genetics. We'll be doing genetics in the next session, guys. One short video, that also genetics. After genetics, we will be doing the second part of it, that is your applications PYQs. Okay, guys, this is Vamsi signing off from Biolab India. I hope no one has joined the session, but no problem. You please go watch this. You please watch this session. It is very useful. We have done many PYQs. We have done 50 PYQs in the biotechnology process part, and you can see that every year minimum 3 to 4 questions are being asked from this topic. Minimum 3 to 4 questions. In 2017, which is not too long away, it is not too far away from us, there are 4 questions which are asked. Those 4 questions are very easy, so that we can put it in 1 minute. So please do practice more. If you have some other books also, please do practice more. Okay, if you want, let me do. If you want more and more reference books, if you want more and more questions from reference books, please let me know in the comment section below. I'll be bringing many reference books, and then we'll be doing this. Okay, then we'll be doing this. Okay, now this is Vamsi signing off. Once again, I am telling you, please do them. Please visit this lecture. Please watch it. Please do them. I know you have asked for a recorded lecture, but my thing is, my notation is, if it is a live lecture, many of you can join it and then interact with the answers. It would be much better, right? So yes, no problem. I'll try next time my best in order to inform you in advance, well in advance, that we are having a live session on this topic, so that you can prepare and you can come. You can arrange some time for me, and then we will be doing our best. So thank you, guys. Thanks for watching the lecture. Those who have watched in between, those who have come in between, also thank you for coming in the between. I hope you are with one question. No problem. Have you a great day further, guys. This is Vamsi signing off. Thank you. Bye bye. I'm gonna